Introducing Research Center for Arkeometri. Established in 2002 as the part of the Research Organization for Archaeology, Language, and Literature. Research Center for Arkeometri is applying and developing scientific techniques and methodologies to Indonesian archaeology. Our mission is to reveal various ways humans engage the material world and the natural processes involved in this relationship. Among us are experienced and passionate scientists. Collaborate in an integrated ecosystem of research, technology, and innovation. Bring out interdisciplinary studies, such as paleoanthropology, investigates human remains using the bioarchaeological approach. Geoarchaeology, applying the earth sciences approach. Archaeobotany, identifying plant remains in archaeological contexts. Pyrotechnology, examining artifacts produced by fire technology. Traceology, interpreting the traces and residues left on artifacts. Digital archaeology, emphasizing digital data acquisition. Arkeometry plays an essential role in providing a robust scientific foundation for understanding our origins and past civilizations. Elucidate the historical background of the unity in diversity as our national identity. Deliver science to the world of Indonesian archaeology.
So hello everyone, good afternoon from Papua and guten Morgen Selina, may I call you Selina? Okay, so yeah, we are really grateful to have you here, and I think it's early morning in Germany at this moment, right? So okay, so my name is Marlene, and I will be a moderator for today's lecture series on the topics about prospect for Southeast Asian in the future on ancient DNA research, which will be presenting by Dr. Selina Carlhoff from Max Planck Institute, Leipzig, Germany. In the past, Dr. Selina led the research on the ancient DNA analysis from individual called Bese that was encountered at Leang Paninge site in South Sulawesi, together with the team from Hasanuddin and Griffith University, Australia. And I just want to remind all of you about the result from genome analysis of Bese that was dated about 7,000 more, if I'm not wrong, years old, and which is surprisingly express a substantial East Asian ancestry component, and which a mixture of Denisovan gene flow, indicating that East Asian ancestry present in Wallacea much earlier than Austronesian expansion. And this discovery has overthrown our common understanding about Southeast Asian human peopling and history, then shows that the peopling of Southeast Asia was much more complex than has previously been appreciated. And just for information, in the last 2 weeks, we have, or 3 weeks ago, if I'm not mistaken, we had Prof. Matsumura from Sapporo University, who is known for his work on morphological analysis. And according to his work, that he proposed that a theory of 2 layers of human occupation in Southeast Asia. In contrast to this hypothesis, today we will hear about the DNA topic, especially from the result from Indonesia and in Southeast Asia generally, which seems to be pointed to multiple layers of human occupation based on the DNA study in this region. And for the most important thing, we will learn about what DNA is and how its future prospect in archaeological research in Indonesia. And we have about 1 hour and 45 minutes to her and discuss about the DNA topic. So, but before that, I think we have open remarks from Dr. Yogiswara, Heri Yogiswara to open our lecture series for today. Silakan, Pak.
Okay. Thank you, Marlene. Good afternoon for Indonesian participant and very early morning, I heard, for Selina in Germany. I would like, first of all, I would like to introduce the National Research and Innovation Agency, BRIN, to participants who not really familiar with that organization. Actually, BRIN is a new and the only one research and innovation institute owned by government in Indonesia, and BRIN has 12 research organization and also composed of 85 research center. Maybe I don't know, smaller or bigger than Max Planck. Yeah? And then one of research organization, namely archaeology, language, and literature. Maybe the name is quite stranger for you, how archaeology, language, literature can be in the one research organization, because that in part in our history join the organization related with archaeology and also language and literature. But actually we are consist of 3 research center related with archaeology. One of them is arkeometri to help this meeting, and the 3 research center related with language and literature, and also 1 research center related with heritage, civilization, and religion, something like that. That I think the brief introduction about our organization. If we talk about our public lecture today, because actually Indonesian archipelago, or sometimes we call that Nusantara, is a really living laboratorium of the very rich bio and cultural diversity, and also diversity of the human genetic from the various period, from the prehistoric until the modern time. I think that we are really rich of that. And also through research, we found many human remains, skeleton, that's content of the DNA of the ancient DNA. And also, BRIN now is developing special laboratorium for ancient DNA. Actually, I and Pa Sofan visit the laboratorium already, all already there, the machine, tools, room, but I think not set up already. Yeah? We need more time to set up everything. And then, of course, if we have already with this laboratorium, we need the collaboration and also transfer of knowledge, like with Max Planck Institute, the place you are working now. We are really hope also to really transfer of knowledge in this public lecture. And of course, enjoy listen the lecture. Thank you.
Ya, terima kasih banyak Pak Heri, dan saya juga menyapa sebelum kita mulai semua, saya juga menyapa, I'm sorry Selina, I combine Indonesian and English, so I'm sorry for that. So saya juga menyapa, ya tentu kita sudah hadir Pak Kepala Pusat Riset Arkeometri, Pak Mas Sofan Nurwidi, kemudian Ibu Asi, kemudian ada dan banyak lagi ini di bawah Mas Adi dan lain sebagainya dari PR Arkeometri, dan juga dari PR lain dari ORR Bastra, dan juga jika ada dari hadirin yang hadir dari PR yang lain, selamat datang. Terima kasih sudah meluangkan waktunya semuanya. Semoga kita selalu sehat. Dan saat ini ya di dalam seminar atau lecture series kita pada sore hari ini akan berlangsung dalam bahasa Inggris, walaupun begitu nanti jika ada bahasa Indonesia dalam hal pertanyaan mungkin bisa juga diajukan, nanti saya mencoba dengan bahasa Inggris saya yang terbatas untuk mentranslate-nya. Okay, we have about 1 hour and 45 minutes to go and to hear and discuss about the DNA topic. So from now, Selina, the stage is yours.
Thank you very much, Martin. Let me share my screen and yeah, we can get started. Yes, hello everyone, also from my side. Right now it's like 10 in the morning, so not super early, but I appreciate you all coming to this talk on a Friday afternoon or evening. I'm very honored to be here, and thank you very much to the Department of Arkeometri and to Bryn generally for inviting me to give this talk. I'm very excited to introduce to you our work in ancient DNA and the potential we have in Southeast Asia, and to discuss and answer all of your questions at the end. So just first a bit about myself. So my name is Selina Carlhoff. I am a PhD candidate at the Department of Archaeogenetics at the Max Planck Institute for Evolutionary Anthropology, which is in Leipzig in Germany. And during my PhD, I've been mostly focusing on introducing and applying archaeogenetic methods in different projects in Southeast Asia, including in Indonesia. 2 of my projects were, and then I have another project in Thailand and in Taiwan. And I've really come to really appreciate the region and all our collaborations there. And I'm very happy to also talk a bit about my results from my PhD today. So in this talk, I first want to talk a bit about what archaeogenetics is and what makes it so challenging and difficult compared to modern genomics, and what kind of methodology do we apply to overcome these challenges and still get our really interesting results. Then, of course, it's important to also understand what kind of questions we can address with archaeogenetics studies, and also might be of particular interest to you what kind of questions, particularly in Southeast Asia, could be illuminated by archaeogenetics of course, in collaboration with archaeology, anthropology, and linguistics, and all other kinds of fields that look at human history. And to illustrate how all these different fields kind of come together to give us insight into the human past, I want to introduce the paper that Marlene already mentioned earlier, where we studied the early human settlement of Wallacea, or just specifically Sulawesi, and studied the DNA of a 7,200-year-old individual. So to get started, archaeogenetics as a word is just made out of 2 different words. One of them means ancient in Greek, and one of them refers to genetics as the study of heredity. So broadly speaking, archaeogenetics just simply refers to the study of ancient genetic material, or ancient DNA, or aDNA. And because DNA includes the genetic instructions for the development, functioning, growth, and reproduction of all known organisms and a lot of viruses, when we combine our knowledge of ancient genomics and present-day information, we can understand the evolution and development of a species through time and in many different contexts and environments. So in simplified, archaeogenetics is simply a way to look into the past and what was going on genetically in the past, and then to understand what happened between this point in the past and what we observe in the present, and what this tells us about evolutionary processes or cultural changes, etc. So compared to many other scientific fields, archaeogenetics is still relatively young. So the first really ancient DNA-focused paper was published in 1984, where the team around Higuchi et al., they were studying the DNA of an extinct horse relative, the quagga, which you can see in the picture at the bottom. And they extracted DNA from this museum individual and were able to compare it to modern horses and understand how they were related in the past. And all of this was kind of made more probable by the development of polymerase chain reaction, which is a method where DNA can be replicated exponentially. So even if we just have a very small amount to begin with, when we use polymerase chain reaction or PCR, we then have a lot that we can study, and it's easier to understand. So after this, many people started looking into ancient DNA and studying extinct organisms and all this. But in the 1990s, many of these studies became controversial because sometimes it wasn't clear if we were really studying authentic ancient DNA, or sometimes it wasn't possible to actually reproduce a result in a different lab. So researchers came together and agreed on some very strict control measures and methodologies to ensure that the DNA that we are looking at is actually ancient, and that the results we have can be reproduced. And among all of these controversies, and under very strict safety precautions, in 1997, the parts of the mitochondrial genome of the Neanderthal were sequenced and published, which formed the first genetic material that was studied from a human relative. And then in 2005, another technological innovation, this time next generation sequencing, just allowed for being able to study many more DNA sequences in a much shorter time and for with much less costs. So this kind of led towards the ancient DNA revolution, which the large increase in studies and labs and papers and genomes published since around 2010, 2015 has been called. And if you look at the number of publications that mention archaeogenetics or ancient DNA over the past decades, you can see really how the field has taken off within the last 10 years. But to understand why it was so hard to get the field off the ground and why it's still quite challenging to actually extract DNA from deceased organisms, I want to give a very brief introduction into the structure of our DNA. And in humans, it's the DNA is commonly, I mean, most people I think are aware that they occur in these chromosomes, but the chromosomes are technically only like a very condensed and like wrapped up version of this very long, one very long strand of DNA that is made of this double helix where sugar phosphate backbone is connected to a partner through the so-called nucleobases. And of these nucleobases, there's 2 different types, within in total 4 different nucleobases, where on the one hand, we have adenine that always occurs opposite of thymine, and guanine that always occurs opposite of cytosine. So the entire DNA and the whole genetic information of humans, mammals, like all other animals and many bacteria, and also some viruses, is made of basically only of these like 5 ingredients. So it's just a different combination of these nucleobases that determines what we look like, what kind of functions different cells have, what we inherit to children, and everything. This is quite amazing, I think. And while we are alive, this, because it's so central to everything that we do, the DNA is constantly repaired because it's such a long molecule. But of course, when we die, all these repair mechanisms are turned off, and then we see that the ancient DNA, that the DNA is actually quite vulnerable to chemical alteration, and is particularly, even like in presence of water, hydrolysis is one of the main mechanisms that damages the DNA. And what happens very often is that the connections between the backbone and the nucleobase are destroyed by the presence of water, and this leads to so-called depurination, which means that the nucleobase, in this case the guanine, is lost into the environment. And because of this gap in the strand often breaks. And this strand breaking leads to the very characteristic pattern that we observe when we actually look at all the fragments of DNA that are present in a sample. So usually the DNA is this very long strand of several billion base pairs actually, but because of this depurination, the strand is broken up into smaller parts, which are mainly between like 30 and 70 base pairs long. And also these small fragments are still vulnerable to more hydrolytic damage. And what often happens, particularly at the ends of these fragments, is that the cytosines that are now exposed, because they don't have a partner anymore, are, again, by hydrolysis, are turned into another type of nucleobase, the uracil, which usually only occurs in messenger RNA, so during protein production in the cell. But so now there's kind of the wrong base in this position. So when we start analyzing the sample, we replicate it many, many times with the polymerase chain reaction, and opposite the uracil, we will get an adenine binding to it. And during another round of replication, this is then split again, and the complementary strand created, we will then, opposite of the adenine, of course, get a thymine incorporated. So in the end, in the same position that originally had a cytosine, we will then, when we sequence it, actually observe a thymine. And this so-called C to T misincorporation is also known as DNA damage. And when we actually look at all the different fragments, we can see that this rate of damage or of C to T misincorporation is particularly enriched towards the end of these fragments. But of course, the short length and this chemical alteration of the DNA make it more challenging to actually retrieve and analyze the DNA that's present in archaeological specimen, and require specific laboratory procedures to actually be able to pick them up and study them then also later during analysis on the bioinformatics side. And so DNA is not only very short and chemically modified, it also, because it is, for example, has been buried in the ground for several thousand years, there's also a lot of other DNA present in the bone. And mostly that's bacterial DNA from bacteria that live in the soil or that have colonized the bone after the decay of the individual. There's other species present. There's flora and fauna. So the actual target DNA here that you can see in the middle is really the minority compared to all the other DNA that we find here, and we call this contamination, because we're looking, we want to identify the target DNA among all this other DNA. So to address all of these problems, so the short length, the chemical modification, and the contamination, there are very specific laboratory methods that we've employed. And one of them is that we try really, really hard to avoid further contamination of any of the samples. So the work that we do occurs in a so-called clean room, which is exclusively only used for ancient DNA, and through positive air pressure, everything is tried to be kept outside. The whole building is irradiated with UV during the night to destroy any leftover DNA. And also when you work in the clean room, you kind of look like this astronaut in a full bodysuit with a mask and 3 layers of gloves that you have to change every time you touch something, and usually also goggles. And also all the material in the laboratories themselves is constantly cleaned with bleach or specific DNA destroying cleaners, and after every sample, to make also sure that we don't transfer any DNA from one individual to another, or of course don't transfer our own DNA into the sample, because we want to study the people in the past and not always sequence ourselves. So that's already many of the safety precautions that we take before even considering touching the bone at all. But the specific methods for actually extracting the DNA start when we first look at the bone, and in our case, we usually look at the petrous bone, which is a part of the skull. It's actually the bone where the inner ear is located in, or teeth. And these are both materials that are very hard, so they've been shown to contain a lot of DNA compared to other parts of the body. And for both the petrous bone and the teeth, we then use a dental drill to drill a very small amount of material from the bone. This is usually around 50 milligrams, but nowadays you can also use from 10 milligrams, 10 to 50. That's usually what we try to go for to be able to have a decent amount of DNA present. And this bone powder, excuse me, is then we add these chemicals to the bone powder that break open the structure of the bone and remove everything of the cell that's not the DNA, hopefully, before we then build these so-called DNA libraries, where in the end we hope to achieve that only the DNA is now left in the suspension, so in the liquid portion of our sample, and then what we often use, especially if there's only little DNA present, is the so-called in-solution capture enrichment, where the DNA fragments bind to specific positions. They bind to prepared probes, and then specifically enrich certain parts of the genome that we know are informative about population history and the evolution, to be able to increase the amount of DNA that we find. So while the petrous bone and teeth are the most common sources for ancient DNA, there's of course many others that are possible, since DNA is present in all parts of the body. So, but depending on your research question, you might want to target different sources of ancient DNA, different parts of different skeletal material. So also other bones are of course possible. And then there's also dental calculus, which is calcified plaque on the teeth, which can also be informative about what the person was eating and the microbiome, but also is very good at preserving pathogens. So also studying maybe what kind of diseases the person might have had. Dental calculus is very, very good for this. Skin and hair and are also very good sources of DNA if they are present. And if you want to study the microbiome, also coprolites might be interesting. And one of the new research groups at my institute is really looking into understanding DNA from sediment directly. So if we don't have any bones present, we can also extract the DNA directly from the sediment. But so far, this is still quite challenging. And the genetic material that we find is very short. So, but they're developing also new methods to try to maximize the yield that we get from sediment, and how this might be very useful to understand regions where we don't have burials, maybe understand the settlements in comparison to burial areas. And there's always new developments in the field. Only yesterday, a new paper was published by some colleagues in another department at my institute, who were able to show that also DNA is also present in bone jewelry that people wore. So even without actually drilling into the bone, they were able to wash it off the surface in a way. And this is of course also very exciting, and understanding more technologies that really reduce the destruction of the skeletal material is, of course, of very high importance. So what are actually all the things we can study with ancient DNA? And since I focus mainly on humans, I will start with those. And from previous studies, we have now know a lot more about the relationships that humans have with other hominins. For example, you can see here the tooth is from the so-called Denisova, which was a hominin that lived in Eastern Eurasia, and we only really have teeth and a mandible somewhere and some finger bones of this individual, of this hominin. So anthropologically, that's very hard to understand. But from the DNA, we could show that the Denisovans were related to the Neanderthals we find in Western Eurasia, and that similar to how the Neanderthals admixed with modern humans during the Out of Africa event, they were able to show that also the Denisovans actually have left some of their ancestry in the modern human genome, particularly of groups that nowadays live in near Oceania. So really understanding how these connections between different hominins took place, and kind of is one of the really important questions of ancient DNA. And also within humans themselves, many questions on how people in different regions were connected, if there was exchange, how cultural and archaeological changes, if they were accompanied by the transfer of genes or just cultural diffusion, is really one of the questions that really interest many people. Particularly here, I also have an example. It's from a publication that studied the expansion of the Yamnaya culture from the Central Eurasian steppe to the east and the west. And during this expansion, they affected and they changed the local gene pools forever. And also on a smaller scale, we are able to understand study the structure and relatedness of people at a site. And in connection with anthropology, it might show us something about how these people lived in the past, what kind of ideas about death and kinship groups they had. So from the really large scale questions regarding human evolution and hominin evolution as a whole, to very small scale specific local questions, there's many different things we can address with ancient DNA. And also not to forget that we can also study the DNA of animals. And here, of course, we can focus on how human activity has impacted the gene pool or the evolution of different animal species or groups. So, for example, how domestication has shaped the genome, and maybe how what people in the past were selecting their domesticated animals for. And also generally, of course, understand the evolutionary history of a species or a group of species, and their distribution and extant relatives, how they relate. And even the very smallest living organisms, such as microbes, also contain, especially like most bacteria, contain DNA that we are able to study. So if they occur if they are pathogens and occur within an individual, we can understand if this individual had any disease and if they maybe were the cause of death. And also how this pathogen evolved over time, and maybe when it became so pathogenic to humans. So a lot of my colleagues also study Yersinia pestis, which is a bacterium that causes the plague. And by studying how Yersinia pestis looked like during the Black Death in the Middle Ages, or the Justinianic plague in Roman times, or the early Bronze Age spread of the plague, understanding how the bacterial genome has changed between these different epidemic outbreaks has really shown us a lot about how bacteria evolve and reproduce, and how they then also affect other species such as humans. And when we look at the bacteria are not just pathogenic, but also a lot of bacteria live within all humans, on the skin, in the mouth, in the gut, everywhere. They're always with us. So from looking at the microbiome in the past, we can also understand what people ate in the past and how the microbiome might have changed, what happened, and how it compares to modern day, and understand how all the different bacterial communities work together. And in Southeast Asia, particularly, there's still a lot of open questions, but so far, there have only been very few archaeogenetic studies. And all of these have only focused on a few individuals each. So in this map, I've plotted some published and unpublished sites where and how many individuals have been able to yield ancient DNA from this site. And you can really see that it's very widespread geographically. So we have like a few sites from Thailand and even from Indonesia, which is a huge country with a very interesting and important history. There's only really these two studies that we published last year and the year before, and all of them have around one or maybe two individuals from the same site. So still, we don't have a lot of comparative data to understand really the fine-scale genetic structure in the region. So there are still, you can see a lot of gaps where no one has studied ancient DNA before. And one of the reasons I suspect is, of course, probably that due to the tropical climate and the high humidity, it seems that DNA decays very rapidly in tropical regions. We also see a similar pattern, for example, in the Caribbean, where my previous supervisor now works in. So the challenging preservation conditions, maybe you've also observed this for bones, is really one of the large obstacles, I guess, here. And of course, it would be amazing if we had, like I said, some more dense geographical sampling. And what it would also be really interesting is to understand in one region, really through time, how things have changed and how different cultural influences, how it changed the gene pool over time. So because the archaeogenetic field, especially compared to other regions of the world, where, for example, in Europe, there's large studies published with more than 100 individuals each, or 100 individuals from one site. I think in Southeast Asia, the field is really only beginning. And I think that's also an amazing opportunity to make sure that the field that we develop is really collaborative and involves everyone who should be involved. And I believe particularly because, of course, we use destructive methods. We don't need a lot of bone powder, but it's still, we have to drill into the bone, and bones are obviously spiritually and also, just for society's important heritage. I think, I believe that really the development of these methods and of any archaeogenetic study, particularly in regions such as Southeast Asia, have to be in collaboration with local scientists, local governments, and communities to ensure that all stakeholders are informed and have a say in what happens to their heritage. So there's still a lot of, like I said, a lot of room for more research. And here I want to just give some examples from like a very large continental to local scale, where I think that would be interesting questions that we can address with the help of ancient DNA in cooperation with many other fields of science. And so on a continental scale, of course, Southeast Asia is a very important, and especially Indonesia, a very important crossroads of human settlement, and very early human expansion into the region clearly had to go through Indonesia to reach Papua and Australia, and then later the far reaches of Hawaii and Aotearoa. So really, it's a very important region to understand these large settlements and also understand how the diversity and what was happening in the past, what kind of people lived there, how they interacted, how much exchange was there once the islands were settled. And along these lines, of course, we're also interested in understanding how modern humans and archaic hominins interacted with each other. So not just the Denisovan, which I've mentioned before, but also, of course, in Southeast Asia, there are local archaic hominins such as Homo floresiensis or Luzonensis, where it would be really interesting to understand if they and how they interacted with humans, if we see any signal of interactions between the two different groups, and what this also means then for the groups in the past. And if we go closer in time to the present, then another large event, of course, in the region was the spread of farming-related groups on the mainland, coming from what's nowadays southern China, but also in the islands with the Austronesian expansions, where we know that they also introduced, new groups arrived in the region, and we can see that they admixed with local settlers, and how this really happened, how these interactions took place, and how over time the genetic makeup has also changed. And at the very local scale, with ancient DNA, we are also able to really zoom into a site and understand maybe if there are any differences between where people are buried, how this relates to their genetic relatedness to other people in the site or other people at other sites. and maybe also what it tells us about individual mobility. So when we don't see a whole influx of a new genetic signature in a region indicating that many people came at once, but how also all the trade networks that historically existed in the region, how they might have also shaped the histories of individual people that we find, and how they connected between different regions and different parts of the continent. But yeah, these are just a few examples to give you an idea of what kind of questions we can address with ancient DNA. I'm happy if you have any suggestions or questions to discuss at the end if you have any ideas. And finally, I want to give you a little insight in the results we can get when we combine archaeology and archaeogenetics, and want to introduce this paper that was published 2 years ago now, nearly, where we focused on studying the settlement of Wallacea through this genome of a Middle Holocene hunter-gatherer from Sulawesi. And of course, when we think about the settlement of the region, this goes pretty much all the way back to around 40,000–50,000 years, where we have, of course, archaeological evidence that humans were already present in the region, either directly through skeletons, and I've indicated some key sites here on this map, but also from the cave paintings, for example, on Sulawesi, for example, that are 45,000 years old, and also other indications of human influence on the environment, on floras and other parts of the archipelago. And it's particularly an interesting region, and because Wallacea, so Sulawesi, the Moluccas, and parts of the southern Indonesian island chain are deep sea islands. So they were never connected to the mainland, which even at the lowest level of the sea connected Thailand and Borneo and Malaysia on the one hand to form Sunda, and also connected Australia and New Guinea to form a large continent called Sahul. But because the Wallacean Islands were never connected to either of these large land masses, it's clear that humans had to cross the water to reach them. And I think that's what makes it really, really exciting, because it's not an obstacle that humans had to cross before, but we also see that they must have crossed the whole region pretty rapidly from the dating of the archaeological sites that we find. So it really illustrates something about humans in the past and how they interacted and how they viewed the environment. But of course, also 40,000 years in the past is a very long time ago. And in Wallacea themselves, as far as I'm aware, the oldest skeletons that we have are from the Ilo Islands around 13,000 years ago. So yeah, really understanding how humans and when and where they crossed through Wallacea, if it was along the northern route or the southern islands, it's really still big questions. So this kind of the archaeological background and the human settlement of the region has also been looked at from the perspective of present day people that live there now. So studying the genomes of people who live in the region now to try to say something about the past. And one of the things that's very, very interesting is that we can really trace like the splits between the like the genetic differentiation between all other Eurasians and people who nowadays live in near Oceania, all the way back to around the time of the first settlement of the region. So between 71 and 52,000 years. And then if we even differentiate between Papuan and Aboriginal Australians, we can find that their genetic ancestors diverged 37,000 years ago, which is an incredibly long time in the past. So also with a few initial ancient genomes, we've been able to kind of populate this tree a bit more. So in 2018, a group from Copenhagen and was able to produce ancient DNA from a 7,000-year-old individual associated with the Hoabinhian technocomplex from Laos. And they could show that this, while this individual lies on the branch of present-day Neo-Oceanians, they are more closely related to present-day inhabitants of the Andamanese Islands. While another ancient genome from around this time, from southern China, actually lies on this other Eurasian branch. But what happens in between and where actually these the groups split and how they interacted in Wallacea themselves, because we didn't have any genetic data, was unknown. And this is also more interesting because of this Denisovan ancestry that nowadays we find in Papuans and Aboriginal Australians. Around 3% of the genome is related to the Denisova. But nowadays this ancestry only occurs across the Wallace line, so past Wallacea. And even the ancient individual, the Hoabinhian associated individual, did not have any Denisovan ancestry. So it's also still an open question where this admixture between Denisovan related groups and all the ancestors of human groups in the regions, where this happened and how it took place. So to summarize, our question was to address, of course, the scarcity of human remains in Wallacea specifically, that date back to the Holocene, like the early Holocene or mid-Holocene. Then we were interested in understanding the crossing routes across Wallacea, and generally just to get an insight into the genetics in ancient Wallacea. And also how and when the Denisova ancestry was introduced into humans that were coming through the region. So I was very fortunate to work with researchers from Hasanuddin University and Griffith University in Australia, who were excavating this site in southern Sulawesi called Liang Paninga, which is a rock shelter. You can see a picture here, and which they found a lot of Tualayan associated material culture. So this is a technocomplex that's very specific to southern Sulawesi and occurs over a long period of time, but only in Sulawesi, in southern Sulawesi. And they have these very distinctive stone tools. Here they're called Maras points, which I show a few examples from the site here. And very fortunately, at Liang Paninga, they were also able to discover the first burial associated with this technocomplex. And in the picture, you can see how it was located within the stratigraphy. So it was a flex burial under covered by these large boulders that you can see in the picture. And it was a relatively complete skeleton, which was very exciting. And I was very fortunate to collaborate with them and actually study, try to extract ancient DNA from the petrous bone of this individual. I actually did also the lab work when we were still in the lab. It was very, very special. And fortunately, this individual actually had quite a decent amount of ancient DNA preserved. So we were able to analyze it genetically and compare it to the groups that I introduced in the previous slides. So what we like to do in ancient DNA, when we look at population genetics, to visualize the genetic differences between individuals, we use a so-called principal component analysis, which just takes all the different genomes that you put inside and breaks the differences and similarities down to like these several dimensions. So we can actually look at them visually. So all the individuals in this graph that are closer to each other are more closely related. And so you can see here that on one side we have Neo-Oceanians, and then most like Austronesia, present-day Austronesian groups, and ancient Austronesian expansion-related groups here in the bottom of the graph, and then stretching like a continuous diversity all the way north towards Mongolia and other parts of Eastern Eurasia. And also we have the ancient individuals that I've mentioned before here. So we have the Hoabinhian-associated individuals that are closely related to present-day Andamanese Islanders, and also this individual from southern China, where you can see it falls on a different part of the PCA. So if we now add the genome of the genetic information of the Liang-Paninge individual, you can see that here, similar kind of actually to the geographic position, it falls right between the Neo-Oceanian groups and the Andamanese Islanders. And that was very interesting because you can see that there's no present-day group that we have genomic information for that falls in a similar part of the PCA. So it's a very unique signal and was very, very cool to observe. So from this position, like I've said, we were also interested in the Denisovan ancestry. So if there is some affinity with a little bit of genetic relatedness to Neo-Oceanians, which all have this Denisovan component in their DNA, we wanted to understand if compared to if the Liang-Paninge individual had any Denisovan related ancestry, since again, the Hoabinhian and Andamanese Islanders, they don't have any that we can observe. And when this we measured by finding all the little parts of the DNA in all the individuals that have that originate from the Denisovans. And you can see here in this plot that Papuan and Australian individuals have around 50 megabases, whereas lots of other groups from Eastern Eurasia only have very, very little. And interestingly, Liang-Paninge actually has about half as much as the Neo-Oceanians. So it falls kind of neither with all the other ancient and present-day individuals, nor it's kind of a unique position again. And when we combine all this information in genetics, or even generally thinking about ancestry, we like to think about it as a tree. You know, you all probably seen like family trees also, where you have yourself and your your parents, your grandparents, and maybe even your great-grandparents. And a similar kind of thing we can also do for different ancient individuals and understand who's closer related to each other by the tree structure. So in this case, the tree kind of goes from left to right. So we can see that the most distantly related are the groups that live in Africa. Then there's this differentiations between Europeans and Eastern Eurasians that I've talked about. And we see this differentiation between kind of the East Asian lineages. So for example, the ancient individual from southern China, and these more Oceanian related groups. So we have the Andamanese Islanders, and again the Near-Oceanians here, who are more closely related to each other than to previous groups. And we can also model the Denisovan ancestry that we find in the ancient, in the present day groups now. So we have this introduction from something related to the Denisovan into the ancestors of Australians and Papuans. And when we add Liang Paninga to this graph, we can see that it falls right here. So before Australians and Papuans split, but after the introduction of Denisovan ancestry. But what was very exciting was when we introduce more of these genetic admixtures, we also always observe the signal of additional genetic contribution into Liang Paninga from something related to East Asians, which was very surprising because, of course, the distance between the mainland and Sulawesi is extremely great, and it's for now also archaeologically there's no precedent for this, and we don't quite understand what this means yet, but clearly it shows that there were more connections between even mid-Holocene groups in this whole the whole continent, and it'd be very exciting to compare to other ancient genomes from the region from similar time periods to see how they were connected and how they interacted already during the very early settlement of the region. So to briefly summarize, we were able to study archaeologically and genetically the first Tualayan-associated burial and produce the first ancient DNA from Wallacea from this individual. We could show that this individual was related to the ancestors of Near-Oceanians, and that it does contain Denisovan ancestry, but only about half of what we find in present-day people. And this might be connected to this large proportion of some deep East Asian ancestry. But yes, like I said, it's still a bit unclear what this means for the, like, archaeologically. But yeah, and now to briefly summarize the whole talk, which has been going on for a while, just to remind you again, ancient DNA is a relatively recent field, and the most of the challenges still remain. So ancient DNA is still low abundant, so there's not a lot present. It's chemically modified and fragmented and contaminated, which all requires specific laboratory methods. There has been a lot more archaeogenetic studies coming out, which utilize a lot of sources of ancient DNA, which we can use to look into human, animal, and microbial history. But so far, there has not been a lot of research in Southeast Asia, probably related to challenging preservation conditions. But I think there's a very large potential to combine archaeological and archaeogenetic research to understand the very interesting past of the region. But this, of course, can only happen if we build a sustainable research field based on equal collaboration and community involvement. And you could already see from the author list, all of these projects really are large collaborative projects. And I, of course, want to thank all of my collaborators at all these many different institutions that I've worked with for the Liang Peninga project specifically. And I, of course, also want to thank the BRIN and the Department for Arkeometri for inviting me here and allowing me to give this presentation. I hope you find it interesting, and thank you all for coming today, and I'm happy to answer all your questions.
Okay, thank you so much, Selena, for the fantastic insight about the lecture with the topic DNA for this afternoon. And we have heard about the definition about DNA, the methodology, sources, and how the ancient DNA has transformed our ability to explain the origin of present-day patterns of genetic variation, and also in terms of historical process. And also the ancient DNA allow us to illuminate the evolutionary history of human diversity. And however, related to Selena presentation, the heat and humidity of Southeast Asia tend to prevent DNA preservation. So Selena, I think everyone are excited to share their thought about the lecture today. And this is because, you know, the DNA is one of fancy technique in archaeology fieldwork, especially for us in Indonesia. So here we are, we have, I think, about 3 or 4, maybe more later, I think. So we have many questions here in meeting chat. And the first questions come from Hudi. So here will a geographical condition and difference in climate of each human habitation affect the characteristic of their DNA? So Selena, what do you think?
Yeah, thank you so much thank you so much for your question. I think that's also in my personal experience, that's something that I have observed when working on different sites from different parts of the region, that it can be quite different how DNA is preserved in on different sites, even if they are in the same general geographical region. So there's still, unfortunately, there have been no statistical studies yet how really the microenvironment might affect the DNA and how it decays over time. But this is something that me and my previous supervisor, who works in other tropical regions also in Southeast Asia, but also into the Pacific and in the Caribbean, what we believe is that there must be also another aspect of maybe minerals that occur in the soil or in like some microenvironment that affects how quickly ancient DNA decays here, that might just be accelerated because of the high annual average temperature and high general humidity in the region.
So is that clear enough, Hudi, or do you have any question related about this? Yeah, we clearly heard from Selena about the humidity, the temperature from Southeast Asia is one of the really big problem. And also, Selena, we have the next question from Mega Ayu Alfitri. She is graduate from University Hasanuddin. So I think this is about preservation again, about preservation for Southeast Asia. So she said about what is the recommended preservation protocol for handling findings that are suspected to still contain DNA. So what do you think?
So what we've found is that it's quite helpful when already, when you are in the field during the excavation and you think something might be later used for ancient DNA, that it's really useful if the bone or the part of the skeleton that you're looking at is then only touched with gloves. And that's one of to just prevent that any modern DNA gets mixed into the ancient DNA, and so already out in the field, I think, because I've talked about all the precautionary methods that we take in the lab when the bone has arrived, and we try to make sure that we don't put any of our DNA inside. So if we already start trying to keep contamination really, really low, and maybe try to use gloves, or at least put the piece of material right in a bag or something, that I think would already help.
Okay, so thank you so much. I think I can share it with you, Mega, about this, because I think I got it from Catherine was 2 years ago, Selena, about this, how the Max Planck research and how they handle, how the protocol and the sampling and everything. I think it's a little bit heavy for us, especially in Southeast Asia, because I read about this, we need to provide some cool box, and also other some temperature need to be what is this? We need to be take care about this. And Selena already discussed about this, and for all of us, I will share with you, Mega, about the protocol maybe. So Mas Ruli sudah kan saya tadi bahwa lecture series kita sore hari ini akan disiarkan juga di CPAS YouTube. Minta maaf sekali lagi Mas Ruli, saya sedikit demam panggung ini jadi sedikit kacau. So mungkin kita juga bisa melihatnya di YouTube CPAS. Dan so we have so many questions here. They are really exciting with you, Selena. I hope you're not tired. So we have question from Muhammad Afdal. He is also graduate from Hasanuddin University. He is one of a wonderful student was, I think. And also he is, the question was about Sulawesi challenge is that it does not have volcanoes. So the preservation of bone findings, especially human bones, is very poor to get good DNA samples. Are there other sample options that can be used for DNA analysis other than bones, petrus or teeth? Terima kasih. So, what do you think, Selena?
That's a very good question, and I think so. At least there've been a few like statistical analyses where people were comparing all different parts of the skeleton to see which preserves DNA best, and it still seems to be really that from at least the human skeleton, it seems that petrous bone and teeth are really the ones where we have the highest chance to still find DNA. But of course, if none of this is present, it then depends what you want to look at. So if there's, other bones of animals, for example, present, then you can still, look at the animal DNA if that's interesting to you. Or we're also working on these new methods, like I mentioned in the paper that came out yesterday, about extracting human DNA from animal bone pendants, where you don't need the actual human bone present, but just something that a human has handled a lot. So, for example, jewelry that touched the skin extensively, or they're also trying tools that were obviously used by the humans living there a lot. So that are some options where you can, I guess, indirectly then extract the human DNA from other material, but human material directly. But this is still something that they are trying, and so far, maybe also so far this has not been tried anywhere else but from cave sites in temperate regions. But I think with there's always constantly more technological innovations where people are able to extract more DNA from older bones, from more destroyed bones, and from, so I think it is definitely a challenge if there's no human bones present, especially in tropical regions. But maybe in the future definitely in the future there will be other methods that can also be applied to non-human material.
So I think it's clear enough, Abdal, or do you have any thought about this? I think it's clear enough. Okay, so we still have about 30, my mathematics is a little bit, so we have 35 minutes to go. So apakah ada yang mau bertanya secara langsung? Saya mempersilahkannya sebelum kita lanjut ke chat berikutnya. Ayo, siapa yang mau bertanya secara langsung? Mungkin ada? Mungkin lebih clear kalau berbicara secara langsung, kalau ada yang mau. Supaya kita masih punya banyak waktu, 34 menit. Matematika saya sedikit kurang. 34 menit. Jadi kalau ada yang mau bertanya secara langsung, saya mempersilahkan sebelum kita lanjut ke chat berikutnya. Oke, mungkin masih banyak yang malu-malu mungkin. So we have next question from Ibu Tari. Selamat sore Ibu Tari dari Eijkman. So we know that ancient DNA is highly susceptible to contamination, and ancient DNA has also been fragmented. So how do we know that DNA we're analyzing is ancient DNA, and how do we control and ensure that the DNA is correct? So, Selina?
Yes, very good question. Thank you so much. So what I presented as like the challenges of ancient DNA analysis, so the fragmentation and the chemical modifications on the ends of the strands and the misincorporations are, of course, challenges for us to retrieve all these really short fragments and then later analyze them. But they are also kind of really good markers of actual ancient DNA. So if we had present day contamination from someone who handled the bone or something, the fragments would be much longer. So you can already see if you have a lot of very short fragments, it's more likely that this is actually the ancient DNA that you're picking up. And then also there's been software developed that analyzes all the different reads and identifies these damage patterns, so the misincorporations. And when we then look at the overall picture of all the fragments from one individual, and you can calculate a percentage of the amount of fragments that have this C to T misincorporation at the ends of the fragments, which show you like if that's really high. So we say definitely more than 5% for us to actually look into this individual, that this is definitely ancient. So this chemical change at the end of the read only happens if it was an ancient DNA. So yeah, so by actually looking at the different fragments and how long they are, if they are damaged at the ends, it's also a way for us to then make sure that we are actually looking at ancient DNA. And there's also been many methods developed to calculate how much contamination is in a sample. So they compare this very complicated statistic methods that they use, but they identify the reads that likely belong to the ancient individual compared to the reads that likely belong to like present day person who interfered with the sample. And then you also get a percentage of how much of the sample is actually your ancient individual. So it's about prevention of contamination before we actually sample anything, but also we can also statistically analyze all the fragments after sequencing and identify the ones where we think where the damage is present and that are like really short, to make sure that we are only looking at the ancient reads and not at myself, for example, when I've worked with a sample in the lab.
Okay, thank you so much, Selina. So I'm Rani from Research Center for Biosystematic and Evolution related to chemically altered ancient DNA, C to T. How do we know that it's been altered and how to deal with that? For example, is there a specific analysis involved? So, Selina.
Yes, so this relates to what I just said previously. So we can identify these positions where these misincorporations have taken place, and then from then identify if this fragment is ancient or not. And so there is specific analyses that we can do. There's specific software to analyze this, to estimate how much contamination is in a sample, how much damage is at the ends of the reads. And also when we think about the laboratory, there's also some methods where we can we try to revert part of this chemical alteration. So there are specific molecules enzymes that will go and find where these misincorporations have happened. So where the uracil is now instead of the cytosine, and it will cut out the uracil and put back the original cytosine to reverse the chemical change. And then when we sequence this, it will not have the damage, if that makes sense. So so you can either look at it before after sequencing to figure out how much damage there is. And also in the lab, you can try to revert this damage to kind of remove this signal.
Okay, so ya, saya pikir apakah masih ada yang mau bertanya? Kita masih punya waktu ya sekitar 25 menit. Mungkin ya betul ya Mas Ruli? Masih 20
Betul,
menit.
Mbak.
Ya.
Ya,
Apakah silakan karena kesempatan ini sangat langka mungkin, dan kita juga mungkin dari PR prehistory, prasejarah dan sejarah, ataukah dari PR lain atau dari OR lain, saya persilakan.
Actually, I have one question, if you may.
Oke, silakan.
Now, maybe it sounds funny, but I'm just curious. Is it expensive to make one analysis of DNA for one sample? Yeah, consider. Is it too expensive or moderately expensive? Let let me know your opinion on that. Thank you.
No, I appreciate also the practical questions. You know, while from the top of my head, I don't know exactly, but yeah, I think it also depends, how you if you're able to do the analysis in your own facilities, or if you have to send it somewhere to get sequenced, and pay someone else to do it, pretty much. But I mean, costs are, especially for sequencing, are always have been going down because of all, now it's much cheaper to sequence more, so the individual sequencing is cheaper than it has been in the past. And so I would say, I'm it's actually, as you know, a PhD student, you don't have to do the budgeting, but it's, and I also don't know how it compares, of course, to financing the excavation and everything. That's probably also complicated, but I know that when in my collaborations, we always try to find a way, because I mean, obviously also at the Max Planck Institute, we're financially in a different situation than a lot of archaeological institutes in other parts of the world. So yeah, we try to make it possible if it's interesting. And
Okay, so Celine, so I just want to continue one of I think our leader from archaeology, and Bapak Heri Yogaswara had mentioned that now the archaeological or archaeometry research center we have how to start this. So select this. Also, we have now something like the facility in Cibinong, if I'm not wrong. So about these tools and machine and other things. But what do you think? Do you have any hint for us about how to begin about the ancient DNA research, and how hard actually that as long you've been in Max Planck, and how hard actually it is something like is that really expensive to start from the beginning, or is that we need some technician or other things? Because yeah, I mean, I actually was in Germany for a few years, and that I know a little bit from the laboratory people that the Max Planck, sometimes they have any trouble with the machine and something like this. So I'm just thinking, okay, because this is Germany, they are really known for the technician and everything, but they also have trouble sometimes. So what do you think? Do you have any hint for this, for us to begin for our research about the ancient DNA in Indonesia, for example? And so just it. So.
Yeah, no, it was very exciting to hear that you're setting up an ancient DNA lab. That's very cool. I'm very excited about this, and I think it would be amazing to have, right in Indonesia. And so we actually recently helped set up a lab, an ancient DNA lab, and like from planning and everything in Brazil. One of my colleagues was with us in Jena and in Leipzig for a while, and then went back to Brazil to start building this new building from scratch and buying all the appliances that are necessary and everything. And so I would be happy to help, in any way, or find the people who can really help with all these technical details. But yeah, I think one of the key things that we also have, every day in the lab, is that it's important, to clean everything all the time. And I think especially when you first start setting up, you have to clean everywhere with bleach and water, or like DNA away, or whatever products there are out there right now. So yeah, really all the surfaces, all the fridges, or all the tools, everything. And what also I think helps quite a lot is if you have like specific rooms, or like if you at least only use this lab for ancient material and don't also do the modern DNA analysis in the same place. So it just avoids that, because present day DNA, if you just need some saliva, and you have a lot of genetic material of this person, whereas in the ancient samples, there's only very little. So it's very easy to have for the present day DNA to overpower the ancient samples. So I think that would be something that's, depending on general facilities, I guess, maybe relatively easy to try to, separate and keep also, the storage separate, have freezers to store the extracted material and all this. I know I'm happy to talk to you another time also more in detail if you want, or send you more guidance on this, because I guess starting out is quite scary, and it's a lot of preparation.
Okay, thank you, Selena. So actually we still have I think we still have 10 minutes, right? If is it okay for this? So actually we have Prof. Gono Semiadi. Selamat sore Prof. Dia juga adalah salah satu dosen Prof pembimbing saya di diklat peneliti. Selamat sore Prof. Terima kasih kehadirannya. So he said about any specific method for the extraction. Have you had experience on extracting petrous bone section, and any specific method for this extraction? And the second one, maybe you can just directly answer about this. It is possible to assess the intelligence aspect level from the ancient DNA test you have done. And the third one, I think, I got reminded by Ruli. So what is it about during excavation if you want to use DNA as analysis tools? So you have, if you can pass it for 7 or 5 minutes, you're very welcome.
Sure. So about the petrous portion, it's I mean, it's obviously easiest, sometimes when the skull is already crushed or fragmented, then if it's already, kind of free within the skull, but I've also tried to, like, sometimes you can also kind of, I don't know, it sounds a bit weird, but you can kind of wiggle it out if it's like not completely attached anymore. But it's also possible, I mean, we've also for particularly for Paleolithic humans from Europe, if they're really important samples, we've also before sampled the whole skull. So it's also possible, to take the whole skull into the lab and then try to immediately produce the bone powder while it's still in place. From what I've observed, and also when I was on excavation and sampling the bones right in situ from the burials, it happens, I guess, relatively often that it's already detached from the other parts of the temporal bone. If you're lucky, it's not so difficult. And then if you have the petrous bone itself, there's also different methods. So you can try to drill from the outside to produce the bone powder, but what we also do is cut open a part to be able to really use the hard part near the cochlea to, if that's usually best preserved because it's so dense. But with modern animal skulls, I'm also not sure. I've not actually worked with animal material, but yeah.
Okay, so I hope that everyone satisfied from what Selina says about this. And so, jika masih ada pertanyaan, Selina sangat welcome untuk bisa dihubungi lewat email. Dan mungkin ya saya kira karena kita tidak punya banyak waktu, jadi nanti bisa menghubungi Mas Ruli atau kami di PR Arkeometri untuk mendapatkan nomor dari Selina, maksudnya email, untuk kalaupun masih ada pertanyaan yang belum jelas atau masih ada yang menggantung dan lain sebagainya, mungkin bisa menghubungi kami hingga bisa dihubungkan dengan, disampaikan kepada Selina atau bisa langsung mengirim email ke Selina. So, I think we have enough for today, Selina. This is really fantastic from you, and I think this is not the end from our talk for today. I think there is many collaboration will waiting in the future. So I think we can, this is not the end of this. So for closing remarks, I think this is the time for closing remarks. So I invite Bapak Sofwan Nurwidi sebagai Kepala Pusat Riset Arkeometri untuk memberikan closing remarks-nya. Silakan Mas Sofwan.
Thank you so much, Marlin. I'm sorry if you could hear my voice clearly because in here we have hard rain. Thank you so much, Selina, for your fantastic sharing for us today. We learned many aspects and we could probably develop some question for a specific subject of research. It's very large range, ranging from the early hominin to the modern human. And as we are people who live in the archipelago, we face the three specific geographic. And one of the big question is regarding the population history of this region, including how and when they can and what route they choose, like that, etc. And archipelago is place which should stimulate local adaptation and isolation to produce a high diversity, as we mentioned. And this is the place where we could find the answer or probably more question from this this character of the environment. And as Pak Heri, the head of research organization mentioned before, BRIN, we are preparing for a lab specifically dedicated to ancient DNA studies. And that the problem we face, main problem, for example, as you mentioned, the ancient DNA come from archaeological context, usually is fragmented and altered, and the problem is how to reconstruct the fragmented DNA to a complete one, and how we could identify and how to minimize the contamination. An ancient DNA study is very fast developed. As Selina told us, her research group tried to extract the DNA from sediment. It's very interesting. Maybe we need more supercomputer maybe to identify what kind of DNA inside the sediment. And we wish in the future we could build a comprehensive collaboration between BRIN and Max Planck to develop this field of study. Finally, I would like to thank for our honorable speaker, Dr. Selina Carlhoff from Max Planck Institute, Germany, to give sharing to us this morning, this afternoon. And I would like also thanks to honorable participant, professor, senior researcher, and also our colleague from different research organization, from research organization of health, and also from life science and environment. I hope everyone could learn and benefit from this lecture. And last but not least, thanks to organizer to initiate this public lecture, especially for this group of paleoanthropology. Thank you so much, Marlene. Time and space, I will talk to you. Thank you.
So everyone, everything is set up for today. So this is the time to say goodbye, and we are really thank you, Selina, for your present today, and also we thank you for everyone who's already attending this lecture series for today. Bapak Heri Yoga Suara, terima kasih Pak sudah hadir. Aduh siapa lagi ya? Aduh minta maaf semuanya. Aduh saya tidak bisa mention satu persatu, tapi terima kasih banyak dari ada Bapak Profesor Akinduli, ada dari Hasanuddin University. So Selena, we have Profesor Akinduli here. Also we have Prof Gono dan beberapa profesor di sini. Saya tidak bisa mengatakannya satu persatu karena terlalu banyak, dan terima kasih banyak atas kehadirannya, dan sampai berjumpa di lecture series berikutnya dari So thank you so much. So that's all for today, and so we say goodbye from Papua, from Indonesia. So thank you, Selena.
Okay.
Bye-bye.
Thank you so much.
Thank you, Selina. Thank you, Marlene. Thank you, Pak Sofwan.
Thank
Apa
you
kita
so much,
berfoto
Pak
ya?
Heri, Selina.
Apa kita berfoto? Mas Ruli, berfoto.
Foto dulu.
Tidak usah.
Tidak
Mau
usah.
foto
Oke,
dulu.
so, so thank you everyone. Terima kasih.
Thank you, Selina. Keep in touch with us.
Thank you.
Thank you, Selina.
you for-
Yes, no, thank
Thank
you all so much.
everybody.
If there's any more questions,
Terima kasih.
Thank you, Selina.
Thank you. Bye-bye.
Bye.
thank you. If you have any more questions, just email me. I'm happy to answer everything.
Okay, thank you.
Thank you, Selina.